Hi, my name is Amin Emad, and I'm a PI at McGill University. And today I will talk about our newest work, simulating single-cell RNA-seq data using causal generative adversarial networks, using a method called ground GAN that was spearheaded by two of my students, Yazdan and Abdurrahman. If you're interested in talking to Yazdan, please check out his poster, B350, on Tuesday. One of the motivations of this work comes from gene regulatory networks, which depict regulatory interactions among genes and their regulators, for example, transcription factors. These are useful models that allow us to explain gene expression programs in healthy and disease states. And with the single-cell RNA-seq data nowadays becoming very available, various methods have been developed for reconstructing them from data. However, one major challenge still remains how to benchmark different GRN inference methods that are available. Various methods have been used to form ground truth, for example, from literature or databases of interactions, but these are typically prone to errors and do not correspond to the biological system of interest. Perturbation experiments or ChIP-seq data is another way, but these are usually costly and time-consuming. Again, they may not correspond to the biological system under study, and typically their availability is much less compared to single-cell RNA-seq data. This takes us to using synthetic data, but simulators that generate synthetic data have their own challenges. For example, most of them are focused on generating realistic data and do not incorporate a GRN in simulation, so they cannot be used for GRN inference. The methods that do also have their own challenges. For example, sometimes they do their data or the insights coming from it do not match those coming from experimental data. A study in 2020, Beeline study, found that methods that perform really well on simulated data did perform poorly on experimental data and vice versa, finding a big gap between experimental and simulation. There are methods, powerful methods such as Sergio that incorporate GRN and can be used for GRN inference method. But for example, Sergio requires manual iterative involvement of a user in order to statistically match the distribution of synthetic data to that of real data, and makes it rather challenging. Here we propose ground GAN, which is a robust causal generative adversarial model that generates realistic single-cell data sets, both at steady states and transient states. It captures automatically biological and technical noise of experimental data. A user-defined GRN is given as input and is used to generate the data, so it can be used for GRN inference benchmarking. And also the causal nonlinear TF-gene relationships of the GRN are imposed in the gene expression profiles, and also the gene transcription factor identities are also preserved. If we look at the architecture and the training of ground GAN, we follow a two-step procedure. In the first step, a Wasserstein GAN is trained used in order to generate realistic single-cell data from noise. And here, the GRN is not imposed, but the weights of the generator are frozen from this step and are used as the causal controller in the second step and the architecture of GAN in order to simply generate realistic transcription factor expressions. The transcription factor expressions and noise values are provided as input to different networks that are our target generators. The role of them is to generate the expression of target genes. And the user-defined GRN is imposed in the architecture of ground GAN by making sure that each gene only accepts input that correspond either to noise or transcription factors that regulated in the GRN. And this is how ground GAN imposes the causal relationship between TFs and genes. Here again, we have a critic that tries to ensure that the simulated gene expression profiles match that of the real data, but also we have two auxiliary tasks that make sure that the transcription factor expressions are used by the target generators in order to impose the causal interactions of the GRN. When we compared how realistic our simulated data are to experimental data, we saw a high degree of similarity. And in fact, here you can see the t-SNE plots for a relatively homogeneous data set that has only one cell type. This is on the training data, and this is on the test data, where blue circles correspond to experimental samples and red ones are simulated data using ground GAN. And we see a good mixing of real and simulated samples. We can also quantify that. We used 5 different metrics and compared them against different simulators. And we saw that none of them impose a GRN, and we saw that we outperformed them. And for example, here, the first 3 metrics need to be as small as possible. This one, we want it to be close to 0.5 because this is the AUROC of a random forest classifier trying to say which sample is experimental, which one is simulated. And this MI-LC is a value that we want it to be close to 2 to showcase a good mixing between simulated and real data. And as a point of comparison, we have the positive control that captures these metrics when comparing 2 halves of the experimental test data set. We also applied this to more heterogeneous data sets. For example, here, our data set had 11 cell types. And even though the cell type information was not provided to ground GAN, we did not see any sign of conditional mode collapse. And cells of different cell type clusters were generated by ground GAN, as you can see, for example, here in these islands. Then we ask whether the interactions in the GRN are in fact imposed in the generated data. So we performed in silico TF knockout experiments, one of the capabilities of ground GAN. And when we knocked out a TF and looked at the genes in the generated data, we saw that genes that were not regulated by that TF, their expression did not change. But when we performed statistical tests comparing before and after knockout for genes that were regulated by the TF, we saw a significant change in their expression value. And here you can see the distribution of the adjusted p-value for those genes, showcasing that, in fact, ground GAN has imposed the interactions in the GRN in a causal fashion. Next, you ask whether GRN inference methods can actually pick up the imposed edges. So we formed a controlled experiments. We used GRN boost 2 and used it on experimental data set. And for each gene, we found the top 10 regulating TFs of it. Then we formed a positive control GRN, which we used and imposed by ground GAN. And here we are imposing the interaction between the TFs ranked 1, 3, 5, 7, and 9 for each gene. And then TFs ranked 2, 4, 6, 8, and 10 were not imposed and were kept as a negative control. And then when we applied GRN boost 2 again on the simulated data, we saw that its performance goes quite significantly up. Here we are having an AUPRC of 0.68 on positive control data set, and you can compare it to its performance when applied to experimental data set, this green line. But then when we looked at the unimposed edges, we saw that its performance was much lower than the experimental data, and it was close to random. And this was great news for us because it shows that it is an effective simulator for GRN inference method benchmarking. We also observed that the trajectories and the pseudo-time ordering of the cells generated by ground GAN are preserved and matched the experimental data. We looked at a data set of mouse bone marrow cells differentiating towards different lineages. And we used PAGA to build discrete states and continuous states in this data set. And not only the topology of the graphs built using simulated and experimental data matched, but also the activity of the markers of different cell types of the data had a very similar pattern in a consistent way, between simulated and experimental data. Here we see that for erythroid markers. Here we see them for neutrophil markers. Similarly, we observed that the pseudo-time ordering of the cells were also preserved. So going back to the use case of GRN inference benchmark method benchmarking, we simulated data using ground GAN, and we used 7 GRN inference methods benchmarked in the Beeline study. And here you can see their performance using 2 different metrics. And strikingly, the order of the performance of these methods almost perfectly matched their order in the original Beeline study when applied to the experimental data, and not their ordering based on their synthetic data, which was quite exciting to us because it showed that we have been able to bridge the gap between the experimental and synthetic data for this kind of use case. After that, we were interested to apply ground GAN for performing in silico knockout experiment. This is possible because ground GAN allows us to sample from both observational but also interventional distributions. So we use the data set containing multiple cell types. We generated data, and then after that, we performed in silico knockout experiments where we knocked out the top 3 differentially expressed transcription factors of 4 of our cell types and asked whether that knockout results in less cells of that type being generated by ground GAN. And this was exactly what we observed. Here, for example, even though originally we simulated data in this island, after the knockout of the top TFs corresponding to the cell types here was performed, much less of cells in this region was generated by ground GAN. And we also could quantify this by looking at the distribution of ILC values. So here I introduced ground GAN, which is a causal generative adversarial model that automatically generates realistic single-cell data sets, captures TF gene relationships, preserves TF gene identities, trajectories, and pseudotime, and can be used for benchmarking GRN inference methods in silico knockout experiments or other applications. Finally, I want to acknowledge different trainees in my lab. Check out Yazdan's poster on this talk, but also if you're interested in deep learning for precision cancer medicine and interpretable deep learning models, check out David's poster. And also if you're interested in multi-species out of distribution protein-protein interaction, Joseph will give a talk on Monday, and also check out his poster. Finally, I want to acknowledge all the funders that supported this and other projects in my lab. Thank you, and looking forward to see you in the conference.